Hello everyone and welcome to today's live broadcast, Animal Models for Tuberculosis Research, presented by Yong Cheng, Research Assistant Professor at the Department of Biological Sciences, University of Notre Dame. I am Marjorie Torres of LabRoots and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many of your questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom or center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the accreditation button located in the promotional board at the bottom or center of your screen and follow the process to obtain your credits. Please join me in welcoming Dr. Yong Cheng. I will now turn the presentation over to him.
Introduced by Marjorie. My name is Yong Cheng from the University of Notre Dame. I'm currently a research assistant professor working on projects understanding the mechanisms on the host and pathogen interactions in the context of Mycobacterium tuberculosis infection. Today, I will talk about animal models for tuberculosis research. Before I start the talk, I would like to thank the conference organizers for giving me this great opportunity so I may share my expertise with the colleagues across the world. Tuberculosis is an airborne disease caused by Mycobacterium tuberculosis. Mycobacterium tuberculosis is an intracellular pathogen. It mainly infects macrophages in the lung and survives and replicates in macrophages. Tuberculosis is currently ranked as top of 10 causes of death in the world, above HIV and malaria. According to the WHO report, 1/3 of global population has been infected by Mycobacterium tuberculosis. Among those infected, 5 to 10% of people will develop active TB sometime during their lifetime. The remaining 90% of infected people will have latent TB infection. In 2016, over 10 million people had active TB because of TB infection, leading to 1.7 million deaths, including 0.4 million co-infected by HIV. Tuberculosis is a leading killer of HIV-positive people. In 2016, 40% of HIV deaths were caused by TB infection. TB is a poverty-related disease. A number of factors may facilitate TB spread. The first factor is poorly ventilated and overcrowded living and working conditions, which may provide ideal conditions for TB spread. The second factor is malnutrition and other diseases, particularly HIV, which may reduce body resistance to TB infection. The third factor is limited access to health care. It has been estimated that one person with untreated TB may pass TB on to 10 to 15 people annually. Based on the WHO report, over 95% of TB deaths occurs in low-income and middle-income countries. This map shows you the global TB epidemics. Here we can see TB cases mainly happens in the South and East Asia and the Sub-Sahara areas. These two regions account for over 85% of TB cases in the world. 7 countries, including India, Pakistan, Indonesia, Philippines, China, Nigeria, and South Africa. They are high TB burden countries. They account for a total 65 of global TB cases in 2016. In most cases, TB is a treatable infectious disease. However, compared to the treatment for most infectious diseases, the treatment for TB may take 6 months up to 2 years. This table shows you the recommended combination treatment for TB-sensitive, for the drug-sensitive TB. It includes 8 weeks intensive treatment with 4 antibiotics, isoniazid, rifampin, pyrazinamide, and ethambutol, followed by 18 weeks continuing treatment with isoniazid and rifampin. The treatment success rate for drug-sensitive TB is quite high. It's over 85% in the world in 2016. This table shows you the 4 different recombinant combination treatment for drug-sensitive TB. Here we can see the efficacy of the treatment can change based on the different combination. In contrast to drug-sensitive TB, drug-resistant TB, especially multi-drug-resistant TB and extremely drug-resistant TB, is becoming a public health crisis and a health security threat. WHO estimated there were 600,000 new cases with resistance to the rifampin, the most effective anti-TB drugs. These are first-line anti-TB drugs, of which around 500,000 had multi-drug-resistant TB. The treatment for rifampin-resistant TB and multi-drug-resistant TB is longer with more expensive and toxic drugs. The treatment regimens recommended by WHO for drug-resistant TB will last for over 20 months or even longer time. The latest data reported to WHO show a treatment success rate for multi-drug-resistant TB is around 50%, only 30% for extremely drug-resistant TB. As I mentioned above, tuberculosis is airborne disease. It's caused by Mycobacterium tuberculosis. This slide shows you the pathogenic life cycle of Mycobacterium tuberculosis in the host. Aerosol particles containing Mycobacteria cells is coughed up by the patient with active TB and uptake by the new host. The bacteria is deposited in the lower lung of the new host and recruit the macrophages to the infection site. These macrophages may become infected by Mycobacterium tuberculosis and initiate the new round of macrophage recruitment in the lung, form the granulomas. The granulomas is a well-organized aggregate of cells. These cells include differentiated macrophages and lymphocytes. In the early stage of infection, the granulomas may facilitate Mycobacterial growth and expand the infection by allowing the bacteria to infect new arriving macrophages. As the development of adaptive immunity in the new host, the granuloma may restrict Mycobacterial growth or survival in the host. However, under certain conditions, macrophages in the granuloma may undergo necrosis to form necrotic core in the granuloma. The necrotic core may facilitate bacterial growth and transmission to the new host. For the TB diagnosis, there are 2 traditional methods. One is Mycobacteria culture. The second one is acid fastening for the TB patient's sputum. The left picture shows you the morphology of the bacterial colony on the agar plates. This is the patient's sputum was placed on the 7H11 agar plates. The right figure shows you the acid-fast staining for the TB patient sputum. Here we can see this pink lot bacteria. This is Mycobacterial cells. This slide shows you another sample of the pathology analysis for TB patient lung. Here we can see the cavity in the lung of TB patient. This may from the granuloma. We also can see this caseating granuloma lesion in the lung of TB patient. Under chest X-ray analysis, we can see a light region in the TB patient lung when compared to the health individual. In the TB diagnosis, we also wanted to know what is the histopathological outcome in the lung of TB patient. This slide shows you the histopathological analysis for TB patient lung samples. In the TB patient lung sample, we can see a typical granuloma. This granuloma contains a central core of necrotic lipid-rich material surrounded by foamy macrophages, epithelioid macrophages, and lymphocytes. The pictures on the first row shows you 3 examples of caseating granulomas found in a TB patient lung. The figure A on the first row shows early caseating granulomas in the lung of people with miliary tuberculosis. The figure B on the first row shows you a cluster of caseating granulomas. Here we can see a typical necrotic core surrounded by macrophages and lymphocytes. The figure C on the first row shows an old lesion in a human lung. Here we can see a fibrous capsule and a necrotic center. The figure A on the second row here shows you another example we can find in a TB patient. We can see the cavity formation here. We just mentioned the granulomas in the TB patient lung. So we wanted to know how the edge of the granulomas looks like. The figure B on the second row shows you the HE staining for the TB patient lung sections. Here we can see on the edge of granulomas there are fibrosis. We also can see this region is filled by the lymphocytes and the macrophages. Why the tuberculosis research is so important today? So we need to look at the current issues we have in the global TB control. The first issue is the TB vaccine. Mycobacterium bovis BCG is only licensed TB vaccine. It has been used for almost 100 years old, since 1921. The efficacy of M. bovis BCG wanes over time after vaccination. It has been found BCG has no or little protection against pulmonary TB in adolescents and adults. Additionally, BCG is not recommended for HIV-positive infants because of high risk of dissemination BCG infection. The second issue is TB treatment. As I mentioned above, more and more drug-resistant TB is found in the world, so the new drugs or regimens are urgently required to cure drug-resistant TB. We also need new drugs or regimens to shorten the treatment duration. As I mentioned above, even for the drug-sensitive TB, if it takes 6 months, it's a long time. It will increase the drug-sensitive TB, the drug-resistant TB percentage in the TB patient. So the new drug and treatment may be needed to shorten the treatment duration for the both drug-sensitive TB and drug-resistant TB. We also need new drugs with low toxicity and high efficacy. The third issue is TB diagnosis. For the current TB diagnosis, we use Mycobacterium culture, acid fast staining, as I mentioned above. We also have a PCR-based and ELISA-based diagnosis. We also have a skin test for the TB. However, there's no efficient TB tools we can use to discriminate latent infection and active TB infection. So we need to efficient biomarker so we can use to efficiently discriminate latent TB and active TB in the human. We also need a biomarker we can use to determine treatment outcome. As I mentioned, for the TB treatment, it's a long-term duration. So we need to have a biomarker. We can determine the treatment outcome, but not use the bacterial culture assay. In this case, we may decrease the opportunity for drug-resistant TB occurrence. The tuberculosis research will help us to understand the pathogenesis of Mycobacterium tuberculosis. We also help us to understand the host defense mechanisms in the context of Mycobacterium tuberculosis infection. The tuberculosis study will also help us to develop new TB diagnosis tools, new vaccine, and new anti-TB drugs and regimens. Animal studies is necessary in TB research. This slide shows you the current available animal models. It has been used in the TB research. They include different species of mice, rabbit, guinea pig, zebrafish, and monkey. The monkey is a non-human private animal model in the TB research. To mimic the progress of TB infection in TB patient, it is necessary for animal models to develop human-like granulomas in the lung. For both rabbit and guinea pig, human-like granulomas can be found in the lung after TB infection. Here we can see the necrotic core in the granulomas from both rabbit and guinea pig lung. We also can see this necrotic core is surrounded by macrophages and lymphocytes. However, in early study, it was found the granulomas formed in the MTB-infected mice, there's no necrotic core. Additionally, the bacteria is restricted in the macrophages, is not released to the extracellular environment as we find in the TB patient granuloma. We also can see a typical necrotic core in the granulomas. Here we can see the necrotic core surrounded by the macrophages and lymphocytes. So this is a typical structure of the granulomas we can see in a TB patient. We can see from this figure A here, I just mentioned this typical TB granulomas we can find in a monkey. After drug treatment, a healing granuloma characterized by a central area containing residual epithelioid macrophages with prominent features of involving fibrotic pressure can be found in the lung of monkey. It provides a very good model for the drug study to develop new anti-TB drugs and regimens. Other type of granulomas or lesions also can be found in the lung of monkey after drug treatment. The figure C here can show you a poorly circumscribed areas of interstitial fibrosis with and without prominent area of cellularity in lungs of monkeys after drug treatment. So this one is also the kind of lesion after drug treatment, more similar to the healing granulomas. The figure D here shows you another example we can see in the monkey. So this is a fibrocalcific granulomas with a central area of mineralization surrounded by prominent peripheral fibrosis. So the monkey provided very good animal models, so we can use it to develop anti-TB drugs, even anti-TB vaccine. In the animal study for TB, there's one question is how we determine which animal models we need in the study. This table partially provides the guideline to us how to select animal models in a TB research. Here we can see in rabbits, guinea pig, and non-human primate, we can see the development of necrosis in the granuloma, also the caseation and cavitation in the lung after MTB infection. There's no cavitation can be found in the mouse model. Also, there are few or very rare caseating can be found in the mouse lung after MTB infection and no necrosis. However, because of small size, cost-effective, and availability of immunological agents, mouse are still widely used as mouse model in the TB research. More recently, a new mouse model has attracted the attention of scientists in the TB field. In this mouse model, we can find 3 different types of granulomas after MTB infection. And this mouse model is called C3HEB-FG. They contain 2 different mouse backgrounds. One is the C3HEB, another one is the FEG background. These 3 different types of granulomas I show you here. For the type 1 granulomas, we also call the type 1 lesion here, most similar to classic human TB granulomas, in which they were solid, encapsulated, caseous necrotic lesion. These lesions become evident 35 to 45 days after low-dose MTB infection in the mouse lung. These lesions is originated as a focal accumulation of foamy macrophages interspersed with neutrophils, often proximal to bronchus. The peripheral margins containing abundant, loosely aggregated epithelioid macrophages interspersed with a small number of scattered lymphocytes. The type 2 lesions is similar to a rapidly progressive pneumonia composed almost entirely of neutrophils. Over time, majority of the type 2 lesions had substantial increase in size and contain a central region of caseating necrosis. The central region had evidence of cellular necrosis and cell death. And over time, it will be similar to the type 1 lesion. The type 3 lesion in the C3HEB-FJ mice were cellular inflammatory lesions that were indistinguishable from pulmonary lesions typically found in the Bobc and black cyst mice. The Bobc and black cyst mice, they are widely used as animal model in the TB research. This slide shows you the correlation between the bacterial numbers with the gross pathology of the granuloma in the lung. Here we can see the high number the high number of the bacteria in the lung. They cause the much bigger granulomas in the mouse lung. However, in the low bacterial number, the size of granulomas is much smaller. As I mentioned above, TB is spread through the air from person to person. The tiny water particles containing the Mycobacterium may be expelled into the air when a person with infected TB of the lungs. The coughs, sneeze, speaks even thinner can spread the TB to a new host. Therefore, specific personal protective equipment and the laboratory is needed for TB research. For the animal studies of tuberculosis, BS3 facility is necessary. To mimic the natural infection route of TB in human, animals are infected aerosolly. So this is a recommended infection route for TB research. This slide shows you the aerosol infection device. It is one of the popular device in the TB research. This is a Glas-col inhalation exposure system. It may generate aerosolized bacteria and infect mice. The instrument consists of a large circular tank. This is also called aerosol chamber that contains a circular basket with 5 pied compartments for animals. So this is a 5 pied shaped compartments basket. The aerosol chamber has a heavy lid. This is a heavy lid with 4 locking handles that lock the lid tightly against the heavy-duty gasket. The lid also has 2 UV lamps under its underside, and these lamps come on during the decontamination cycle of instrument operation. The front of the instrument contains a control panel with 4 timers for the various cycles of operation, 2 air flow meters and 2 air control knobs, and 3 holes with clamps used to attach the nebulizer. When the instrument is in operation, compressed air is flowed through the nebulizer and produce a very fine mist of the bacterial suspension, which is then carried by a large volume of air flow into the aerosol chamber here. This fine mist is then inhaled by animals within the basket. After airflow passes through the chambers, the airflow then exits the chamber through 2 HEPA filters and a superheated exhaust stack. This is an exhaust stack, where the air is subsequently incinerated before release. So in this case, we can remove all the bacteria particles before release to the air. After the nebulization process, the machine runs a cloud decay process, in which the aerosol chambers is purged with fresh air. This may also help in drying any bacteria attached to the fur of animals, a surface of basket, and the chambers. A final step is a UV light treatment, in which the UV lamp under the lid comes on and decontaminates the top surface of the basket. Except the techniques we used to infect mice, except the one we mentioned on the previous slide, there's also other techniques we can use, including intravenous injection and respiratory injection. Intravenous injection can be finished through the tailwind or retroorbital sinus. For the respiratory route infection, we can infect mice using intratracheal injection or intranasal infection. For intratracheal injection, the bacteria is injected into the lung of mice directly. Recently, we developed a new technique for intratracheal injection. This slide shows you the assembling of this injection system. There are 3 main components in this system. One is the installation platform. The second one is the illuminator. This is adjustable illuminator, so we used it to localize the trachea of mice. The third is the glass desiccator. We first used 3D printer technique to generate the main part of installation platform. Here, the 3D printer technique is based on the process called additive manufacturing. This technique, a 3D printer technique, it's becoming more and more important right now to create custom designed or sometimes difficult to manufacture products. The file for this platform is available online. It's free for everyone. You can find the link for this file in our paper. After we have released this platform, we also generated two different metal bar. One's a crossbar with mouse inside the hanger. The second is adjustment metal bar. We first insert crossbar into the main part of the installation platform and insert the adjustment bar into the one of the three holes in the platform. So we can adjust the height of the crossbar according to the size of animals in the study. After we have this system assembled, we need to prepare mice so we can inject the mice. We use inhalation as a flooring to anesthetize mice in a heavy glass desiccator, as I'm shown here, a glass desiccator. The mice were then monitored to ensure sufficient depth of anesthesia. After the mice is ready for injection, each mouse was gently suspended by its superior incisors on the mouse incisor hanger and its body placed onto the slot on the bevel. This is a slot on the bevel of the platform. The Scott arc adjustable illuminator with a double gooseneck was positioned near the pharyngo-epiglottic region of the mouse neck. The tongue of the mouse was gently pulled out to the left side using small tweezers and then pushed down with a micro spatula. The trachea was visualized by a percutaneous illumination using the light emitted from the end of a gooseneck fiber optic end. 30 to 50% of samples, this is a bacterial suspension, were gently injected into the lung by intratracheal injection using Eppendorf research pipette and gel loading tips. After injection, the mice were wrapped with a clean paper towel and returned back to the new cage. Mice were monitored until they were fully recovered from anesthesia. The figure E here shows you the yield of bacteria in lung 1 day after injection. Here we can see 90% of bacteria is injected into the lung of mice efficiently. To determine the bacterial numbers or burdens in the different organs, the organ can be homogenized using hand-operated tissue grinder. Here shows you the glass hand-operated tissue grinder. We also can use electronic power grinder. We should be careful when you use this tool, tissue grinder. The experiment should be done in the biosafety cabinet. The homogenization process may generate significant aerosol hazard. So the glass box connected to the safety cabinet is highly recommended, especially for electronic power grinder. Next question is, what assay we can do or perform in an animal study for tuberculosis? We can look at the bacterial input or burden in the different organs by the bacterial culture on the agar plates, or use acid fast staining for the organ sections. We also can look at histopathological change in the organ samples using HE staining or immunostaining. Other experiment we can do is we can perform flow cytometry, like the flow cytometry, RT-PCR, even RNA sequencing or ELISA to look at the immunological change or response in the tissue after MTB infection. Flow cytometry can be used to determine different populations of immune cells after MTB infection and to see which population of immune cells is important to kill bacteria or facilitate bacterial growth in the tissue. RT-PCR can be used to determine specific host or Mycobacterium RNAs in the tissue. RNA sequencing is a more powerful technique. It can be used to determine a host or Mycobacterium gene expression profile in a certain tissue after MTB infection. ELISA is used to determine the cytokine or cytokine production in the tissue. We also can use the blood samples from the MTB infected animals. Using this blood sample, we can look at the different host proteins, such as the cytokines or chemokines. We also can look at the peripheral profile of blood cells in a whole blood of animals to determine which cell type is important to kill bacteria. In a TB biomarker study, we can look at the specific biomarkers we can use to diagnose TB in the future. In an anti-TB drug study, we can look at the drug stability in animal blood. In this case, we may develop more effective or stable anti-TB drugs. The tuberculosis research is important today. As I mentioned above, it will help us to understand the bacterial pathogenesis in a host and also understand the host innate and adaptive immunity response to MTB infection. The study will help us to develop new or more effective TB vaccine and anti-TB drugs and regimens, also help us to find the new TB diagnosis tools. The final goal of the TB research is to contribute to the global TB control and finally find the solution to end the TB in the world. Finally, I would thank you for your attention. I'll be very happy to take any questions.
Thank you, Dr. Cheng, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your questions into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how do you measure Mycobacterium numbers in a stock used in infection?
It's a good question. We do need to know the bacterial numbers in a stock. This will be used for the infection. To determine the concentration of the bacteria in the stock, the Mycobacterium suspension, this we get from the stock, is diluted in PBS in a 10-fold series and then plated on 7H10 or 7H11 agar plates. The bacteria colonies are counted 3 to 4 weeks after incubated at 37 degrees. Before we start the experiment, all the stock is stored at -80.
Thank you for that answer. Our next question is, how many mice may be infected once using Glas-col aerosol device, and is any anesthesia region needed?
In one cycle, up to 80 to 100 mice may be infected when we use a basket with 5 pie-shaped compartments as shown on the slide. But sometimes we also can use a smaller basket. In this case, we can infect less mice. In an infection, no anesthesia agent is needed.
Thank you. Okay, it looks like we have time for one more question. Our last question is, how does Glas-col inhalation system get decontaminated post-infection and sterilize the airflow before release during the experiment?
This is an excellent question because the safety is always the first concern for the researchers when we work on tuberculosis. So when we use the Glas-col system, so there are different steps we can decontaminate. The first is during the infection, before release into the air, the airflow is passed through the 2 HEPA filters, and then a superheated exhaust stack that incinerated any residual particles, as I mentioned on the slide. The second is the after infection, there is a cloud decay step in which the chamber is purged with the fresh air. They can remove any residual bacteria on the fur of animals in the surface of the basket and chamber. After this step, there's a treatment with the UV light. As I mentioned, this UV light is under the lids, the underside of the lid. This UV light comes on, that will decontaminate the top surface of the basket. The third is after infection finished, the nebulizer and the basket will be removed from the aerosol device and autoclaved. The surface of the chambers is decontaminated using disinfectant. Thanks.
I would like to once again thank Dr. Yong Cheng for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.